Hello guys, welcome back to my lessons. So today we're going to do the topic of meiosis, and under this topic what you need to know is the definition of meiosis. You need to know the process of meiosis in animal cells. You need to know meiosis 1, meiosis 2, the significance of meiosis, abnormal meiosis, and the differences between meiosis 1 and meiosis 2. So what is meiosis? This is a type of cell division that results in 4 daughter cells, each with half the number of chromosomes of the parent cell as in the production of gametes. So this process of meiosis, it happens in the ovaries, in females, in males, it happens in the testes. So after meiosis results in those 4 daughter cells that have the chromosomes of a human being, which is 23 chromosomes, those cells will develop to form what we call gametes. So the process whereby those cells are developing to form gametes, that is gametogenesis, a process where gametes are formed. So what happens in males, 4 cells are made during the process of meiosis, and then they develop to form gametes. What are gametes in males? They are sperm cells. In females, those 4 cells that results after the process of meiosis, one cell developed to form a gamete, which is ovum in females. So a process where gametes are formed specifically in males, that is spermatogenesis, and in females, that is oogenesis. So let's move to the process of meiosis, meiosis 1 and meiosis 2. So I want you to take note of what I call PMET. PMET means P for prophase, M for metaphase, A for anaphase, T for telophase. So before this phases, we have a phase that is called interphase, which is the first stage of meiosis where DNA replication takes place. So it means that before the process of meiosis takes place, we have the process of the process of DNA replication, and that phase is called interphase. And then let's look at meiosis 1. We have PMET. What happens in prophase 1? Before we go to prophase 1, let's look at what we call a chromosome. This is what a chromosome looks like, and then it has chromatids. The blue part is a chromatid. The yellow part alone is also a chromatid. So when they come together, we call that point there in the middle centromere. So the whole thing is a chromosome, and then the blue part it's a chromatid. So if we have two chromosomes that are similar, they are equal in height, those are homologous chromosomes. So let's look at prophase 1 of the process of meiosis. So we have meiosis 1 and meiosis 2. So prophase 1 in meiosis 1, that is where the chromosomes, they arrange themselves in pairs, they form homologous chromosomes. And what happens to those homologous chromosomes? The process that is called a process which is crossing over. That is whereby the chromatids of those two homologous chromosomes, they cross at a point called chiasma. That point is a chiasma, and then genetic materials is exchanged between the chromosomes, so they can become identically different from each other. So let's look at the metaphase 1. What happens in metaphase 1? We see spindle fibers. Those lines there, those lines are called spindle fibers, and the orange parts on each side, they're called centriole. So we see that chromosomes align themselves at the equator or in the middle. So how you're going to cram this metaphase, M for middle, the chromosomes, homologous chromosomes, align themselves in the middle of the equator. So what happens in anaphase 1? Anaphase 1, A for apart, the chromosomes they separate, they become apart from each other. So they separate. What happens after they separate? What happens is what we call telophase 1. A nuclear membrane reforms around the separated chromosomes to form new diploid cells, right? So until from metaphase 1, sometimes it's the same as the process of mitosis. And then now what happens during telophase 1 is also what we call cytokinesis. The division of cytoplasm will also have karyokinesis. Karyokinesis, that is the process whereby the nucleus the divides. So let's look at prophase 2. Prophase 2, we have homologous chromosomes again, but crossing over doesn't take place in this phase. Metaphase 2, chromosomes, not homologous chromosomes, but chromosomes align themselves in the middle of the equator. We have the spindle fibers, we still have the centriole. And then we have anaphase 2. Anaphase 2, chromatids, they separate, they become apart from each other. I said anaphase A for apart. So they become separate from each other, chromatids this time, not chromosomes. You need to be very careful if they ask you in the question paper, describe anaphase 1. In anaphase 1, you're going to say chromosomes separate and they become attached, they become apart from each other. But in anaphase 2, chromatids separate. In metaphase 1, homologous chromosomes align themselves in the equator, but in metaphase 2, just chromosomes align themselves in the equator. So let's look at telophase 2. Telophase 2, we see 4 cells there. Cytokinesis has taken place. Karyokinesis has also taken place. We see that there are 4 cells, each containing of haploid cells instead of diploid cells. So let's look at significance of meiosis. There are 2 reasons why meiosis is important, and let's look at them. It reduces the number of chromosomes by half. In other words, from diploid to haploid. This ensures that sex cells have half the number of chromosomes of other somatic cells, so that when fertilization occurs, the zygote formed has the correct number of chromosomes. It balances the doubling effect of fertilization. Crossing over introduces genetic variation. Genetic variation results in offspring that are better adapted to a particular environment and ensures that they will have better chances, a better chance of survival. So let's look at abnormal meiosis. Abnormal meiosis, well, sometimes mistakes occur during the process of meiosis. This can happen in anaphase 1, where homologous chromosomes may not separate. So this is called non-disjunction. It can also happen in anaphase 2, where non-disjunction of the sister chromatids, where there is non-disjunction of sister chromatids. If there is non-disjunction of chromosome pair 21 in humans, that leads to the formation of an abnormal gamete with an extra copy of chromosome 21. If there is fusion between a normal gamete and that abnormal gamete with extra copy of chromosome 21, the offspring will have what we call Down syndrome. So let's look at the differences between meiosis 1 and meiosis 2. Chromosomes arrange themselves at the equator in homologous pairs in metaphase 1. In metaphase 2, chromosomes line up at the equator of the cell individually. Whole chromosomes move opposite to the poles of the cell. In metaphase 2, daughter chromosomes or chromatids move to the opposite poles of the cells. In meiosis 2, 4 cells are formed at the end of the division, and then in meiosis 1, 2 cells form at the end of the division. In meiosis 1, the chromosome number is halved during meiosis 1. And then in meiosis 2, chromosome number remains the same during meiosis 2. So crossing over takes place in meiosis 1. Crossing over does not take place in meiosis 2. Remember, I said in prophase 1 there is the process of crossing over because we have homologous chromosomes. In meiosis 2, crossing over does not take place. Thank you so much for listening. Don't forget to subscribe to the channel, and I would really appreciate